Ribosome footprinting, also known as ribosome profiling or RiboSeq, is a relatively new scientific method that allows researchers to get a more accurate picture of what is going on in a particular organism at a given point in time. More specifically, it gives a picture of which particular proteins are supposedly being produced as a result of the genetic instructions located within a cell. This could be important for a variety of reasons, but we will give one example here. If an employee of a drug company is working on the development of a new drug for the treatment of a disease, he or she needs all the information available to help develop an effective treatment. Knowing which proteins a model organism is producing in response to an exposure to this drug would be extremely helpful in targeting the activity of the new drug. To understand ribosome footprinting, a brief review of basic genetics is in order. The instructions for the development, growth, and maintenance of a living organism are found in its cells. These instructions are encoded by genes, which are made up of small molecules of a substance known as DNA. DNA is a series of smaller units identified by the letters A, C, G, or T. It is the arrangement of hundreds or even thousands of these units in specific sequences that form a code that tells the body which proteins to make. These proteins are responsible for directing and carrying out the functions that are necessary for everyday life. The jump from DNA to proteins involves the molecule known as messenger RNA. Messenger RNA takes the genetic code or message from the DNA and translates it into proteins by the help of molecules called ribosomes. Ribosomes travel down a strand of messenger RNA and read the sequence encoded there. As they read the message, they simultaneously string together other molecules, amino acids, that are the building blocks of protein. This brings us to ribosome footprinting. If we know exactly which messenger RNA has ribosomes attached and how many, we then know which proteins are being produced and to what degree. First, we begin by treating the contents of a cell with a chemical called cyclohexamide, which freezes the ribosomes in place on the messenger RNA. Once this is done, we separate out the messenger RNA with the attached ribosomes, which can also be referred to as polysomes. We separate these from other contents of the cell by passing the solutions through a thick sucrose filter. This is where the really interesting part comes in. Messenger RNA is very unstable because it can be easily degraded by special enzymes called nucleases. Nucleases chew up unprotected strands of messenger RNA. This leaves only the portions of the messenger RNA that are protected by the ribosomes, thus the idea of a ribosome footprint. If we identify the protected messenger RNA segments, then it follows that we can identify the proteins for which these segments encode. To be able to identify these proteins, we still have a little more work to do. Now we subject the segments of messenger RNA that we have to RNA sequencing, which is a relatively new, sophisticated process that gives us the sequence of all of our total RNA. This is done multiple times, which is referred to as deep sequencing. The more we sequence, the greater detail we will generate, and that helps us to know the amount of each footprint that we have. In the end, the segments that we have represent translated protein and are compared to a known reference sequence of the total genetic material of that organism. From this, we can determine exactly what proteins are being expressed from which genes. And because of this, our drug researcher now has much more information to work with, which makes everyone happier and healthier in the end.